mapakita ang aking camera.
Nakikita na po yung screen ko po, ma'am.
Yes po. Wow, ganda.
Start na po ako, ma'am, or mag-wait po ng iba po.
Start na. Okay. You may now start. Lan Coletia.
Carlo.
Ay, wait na po. Inayos ko po.
Okay.
Present ko po ulit.
Yes po.
Yan po. Nakikita na po, ma'am.
Yes po.
Di pa po. Yan, nagsisimula na. Starting ko.
Meron na po.
Meron na po.
Sige po. So, good afternoon everyone. So, this is the result of my bread mold experiment. But first, take a look po dun sa mga na-photograph ko po dun sa while making the experiment po.
Ganda. Amazing ay. Ganda. See? Kita niyo yung floating bodies? Ganda. Ganda. O, diba? Hindi nyo inakala na ganyan kaganda, diba? Akala nyo basta-basta lang yung molds, pero tingnan nyo kung gaano kaganda. Nature's art. Uy, ang ganda, grabe. Sige, sige. Wow.
Wow.
So, yun lang po. So, mold is a common name for many kinds of tiny organisms called fungi. There are thousands of types of molds that can be found indoors and outdoors. Different molds will grow in colonies, living on dead organisms such as decaying plants and animals, as well as non-living materials such as buildings, wood, fabric, and books. Mold need moisture to thrive and usually grow and reproduce spores in damp or moist places. Light and temperature also impact mold growth in different locations. So, ito po yung result of this table. For the first 2 days of the experiment, there were no molds visible both in the dark setup and exposed to light setup. The texture of the slices of bread was smooth and there was no observable color appeared. During the 3rd day, small colonies of black and green molds were barely visible. The molds appeared first on the wet bread, both dark and exposed to light setup. During days 4 and 5, the setup that was exposed to light, the wet one, fuzzy and lumpy colonies of molds grew bigger, but the colonies on dry pieces of bread didn't grow much. In dark setup, larger black and green colonies of molds appear on the surface of the wet bread, while the dry bread didn't grow much bigger colonies. The colonies look lumpy and fuzzy as well. As the day passed during day 6 and 7, as you can see here, the colonies grew bigger and vibrant black, green, yellow, orange, and blue colors appeared on the surface and side of the wet bread in both dark and exposed light setups. In the dry setups, the colonies of molds continued to grow, but at a slower pace. So, conclusion, at the end of the experiment, it was found that molds grow faster in dark, moistened setup rather than being exposed to light. Mold, unlike plants, is not photosynthetic and does not create energy with the use of light. In fact, since sunlight inhibits and even kills mold development, many molds flourish and grow better in dark conditions. On the surface of the bread mold commonly shows as fuzzy blue or green spots. These spots grow black spotty cores if left undisturbed. Based on the color of the molds, black and green, we could see that the common bread mold, Rhizopus tonellifera, was present. The molds need enough water but not in excessive amount, an optimal temperature, and sufficient food for them to grow and thrive. The mold will continue to grow on the bread until it reaches its final stage and the food supply is completely depleted. So, yun lang po. Thank you.
Okay, thank you for that, Carlo. Ang ganda, nakita nyo lahat. Lahat naman ng setup nyo ang gaganda. Nakita nyo yung stock na nagaano sa fruiting body. Eventually, we will have a discussion on that, on paano siya nagmumultiply and paano siya nag-produce ng fruiting body. But you can see how beautiful it is. Na ang akala nyo kapag hindi mo siya tiningnan ng close up, parang ano lang siya, parang anong tawag dito? Parang isang lamp lang na maitim o kulay green or kulay yellow. But if you really look closer, just like the pictures that you placed in your reports, nakita nyo yung mga fruiting bodies really. Yung mga lumps na nakita nyo are actually the fruiting bodies. So very good for that, Carlo. At si Carlo ay umulit dahil nabasa. And trial and error naman really ang pag-aano. But in culture mechanisms, kailangan mo really maggawa ng isang pure culture. Kaso hindi tayo makapagawa ng pure culture dahil wala naman tayo sa laboratory. Kaya pinagkakasya natin ang mga bagay-bagay para matuto kayo at makita nyo na ganun ang kanilang structure. And you can see the fusy body na nakita nyo na ano ito, na nag-i-spread, di ba? Fusy bodies muna, puti muna ang nakikita nyo. And this is you can see the growth basically of the fungal body bago siya magkaroon ng ano tawag dito, mga fruiting bodies. Okay, thank you, Carlo. Okay, Daryl, you're next.
Yes po.
Yes po.
Go da.
Kita po.
Yes po.
Good day everyone. Today I will be presenting the results of my bread mold experiment. Here is a table and the summary of my observations in the 7-day activity. I placed the light setups near the window while the dark setups were placed inside a cabinet. In wet conditions, sa light setup, all replicates show green, white, yellow, and black molds where they all cover almost all the space of the bread. All replicates show similarities when it comes to texture and color. Green and black appear to be bumpy, while the white and yellow molds look fuzzy and smooth. When it comes to dry light setup naman po, replicate 1 and replicate 3 have both green, yellow, black, and white molds, which has the same texture as from the wet light setups. I observed that there was a discoloration in the bread, which makes the bread color brown. While sa replicate 2 naman po, white mold with smooth fuzzy texture and green with bumpy appearance naman yung present dun sa bread. Napansin ko rin na unlike sa ibang replicates and setup, hindi masyadong nag-grow yung mold dito, sa gilid lang po talaga. Sa dark areas naman po, in wet setups, replicates 1 and 2 have green, white, black, and yellow colored molds with bumpy, fuzzy, and smooth textures. While sa replicate 3 naman po, green and white lang yung nag-grow and di rin masyadong nag-expand yung green molds dito. Almost half centimeter lang yung nilaki niya. Sa dry dark setup naman, compared sa dry light setup, yung molds dito nag-spread throughout the bread, pero mostly green lang yung nag-grow. Here are the images taken during the experimentation. To summarize, most setups ay hindi pa visible yung mold sa day 1. Sa day 2 naman po, ma-observe yung maliliit na spots sa gilid ng bread. Then, as the days progress, the molds spread hanggang sa crumb na ng tinapay. Then, doon na po nag-a-appear yung black and yellow colored molds. The spore formation naman po was also observed during the 5th and 6th day po sa green and black molds, which are both bumpy. Napansin ko rin po na nagkakaroon na ng discoloration yung bread. Parang nagiging brownish na yung originally white bread po. And on the last day naman ng experiment, makikita na talaga yung different colonies ng each mold sa buong tinapay. To conclude po, molds can cause harmful effects in our body as it can lead to chronic lung diseases. Molds usually grow in areas with a lot of moisture based po sa what I have observed in the experimentation. I've learned that in order to prevent these molds, dapat dry and clean lang yung surroundings. Yun lang po. Thank you.
Okay, so ganda. So, just to correct you, hindi formation ng spores, but formation ng fruiting body muna kasi hindi tayo nag-observe ng spores. Yung fruiting bodies natin na nakita, yung mga lumps na pabilog-bilog, yun yung ating na-observe dito sa ating experiment. For some reasons, hindi ko ma-turn on ang aking camera at hindi siya gumalaw-galaw. Ayaw niyang magpakita ako sa inyo.
Okay na.
Okay. O yun, nakita niyo na ako. So yun, ang tingnan niyo ako. Wait lang. Kita niyo?
Yeah.
Kasi pumapasok na sa mata ko. Sorry. So, I have to tie it up. Okay, so ang nakita niyo ng mga lumps are actually the fruiting bodies. Hindi po yung spores. Kasi hindi po natin kaya mag-observe ng spores dahil wala po tayong microscope na naandyan sa mga tabi-tabi. Okay, ang susunod po ay si Dwayne.
Go, Jane.
Dwayne?
Po, please po wait. Nakikita po.
Opo, nagsisimula na po.
Okay, thank you. Good afternoon. This is my presentation of our laboratory activity, bread mold. The objective is to understand what mold is and how it grows, observe the growth of different kinds of food molds, and understand how to identify and prevent the mold growths. Procedure is we purchase 12 breads, divide into 6. One is the dried bread, one is bread that we will moist. We will store 3 breads that are dry on the areas that is exposed to light, and the other we will store it in a dark area. Not during day 1 and up to day 4, not much change really. Not much. Only on day 5, some changes are starting to appear, like this image on the right. On the moist bread, you can see that there's spores right at the end of the bread, that green part. Yeah, and again, not much else happened to the bread on the left, the one that is not moist. And up to day 6, more mold starts to appear on the wet bread, the ones exposed by the light. Again, normal, nothing happened on the bread on the left. Now, here's another one, bread that is stored on a dark area. This time, more molds are starting to show up, more greenish with some black color. And now up to day 7, this same setup. This one is from where we store it in. I store it in a light area. More mold starts to appear at the edge, the middle, at the top. Again, nothing happens to the dry bread. And now, this is the one that's stored in the dark area. More variations of molds, green, black, and now yellow. In conclusion, molds thrive on moist conditions. That's why the wet ones is where the mold starts to appear more quickly than the ones that are dry. And yun na po.
Okay, thank you for that, Dwayne. Kakaiba ang iyong resulta. If you remember, sa procedure, nilagay ko, basain lang yung bread. Hindi ko in-specify kung anong klaseng water ang ilalagay natin. Naririnig niyo ako? So, hindi ko ini-specify kung saan galing water, kung mineral water, or anong klaseng water, galing lang sa tap water ang pinangbabasa nyo. You have to remember, sabi ko, na this particular fungi exists in nature. So, pwedeng sa water, pwedeng sa soil, pwedeng sa hangin, actually, yung kanyang mga spores. So, yung paraan ng paggagawa, I decided to really not ask you na ganun ang mga gawin. But if we are in a laboratory condition, kailangan natin gawin ang lahat ng mga bagay na ito. But nevertheless, yung water na pinangbasa nyo sa inyong bread ay merong epekto. Most likely, pwedeng meron din siyang contamination of mold. Plus yung inyong pong lalagyan, pwedeng meron din po siyang hindi nalinisan ng maayos, or kung malinis man sa tingin ninyo ay hindi siya masyadong na-disinfect. Kaya sa laboratory condition, usually ang mga containers na ginagawa for culturing this particular organisms ay ini-sterilize para walang contamination, at paulit-ulit na ginagawa yung pagkukulture dahil hindi nga basta-basta naiiwasan. Kahit na nandun ka sa isang chamber na buong akala mo yung chamber ay malinis and free from whatever, you have to remember na pag nagtatrabaho ka, lahat na ilalagay mo dun sa chamber na yon. And kapag nilagay mo yun sa chamber, pwedeng nandun sa loob ng chamber yung spores ng whatever contaminant at pwedeng makontaminate ang iyong ano. So, dun sa inyong mga setups na pinadala nyo sa akin, actually pwede tayong isolate ng hindi lang siguro mold ang meron dun sa inyong mga ano. Kaso, of course, kung nakita nyo naman, merong isang species na masyado siyang mabilis mag-grow at na-colonize niya ang buo. Kahit na meron ng halimbawa kulay green at kulay white at kulay yellow at kulay itim, paminsan ang bilis-bilis ng itim mag-colonize at na overpower niya yung colonization process nung ibang mga species ng molds na meron. Okay, salamat Dwayne for doing that. So you have to submit your presentation also. Pinag-edit ko na po siya. Hanggang mamayang 7:00 ng gabi, pwede pakilagay ang inyong reports on this sa section na yon. Okay, ang susunod po ay si Phil. Phil? Nandito po ba si Phil? Phil?
Wala po, ma'am. Wala daw po siya, ma'am, na load.
Ha? Wala daw
po
nang
siya na load. Load. Naubusan daw po siya.
Ay, po. Alam niya naman na may klase siya. Ay, Lord. Okay, Phil. Ibig sabihin, walang output si Phil.
Ma'am, by the way po pala, ma'am. Yung kay Angela po, ma'am, di din po, ma'am, makaka-present po siya. Kung pwede po, ma'am, magpasa na lang daw po, ma'am, ng recorded video po, ma'am.
Bakit? Ano daw si Angela? Nasaan din siya?
Wala po sa bahay daw po.
Ay, Lord. Okay, Franz?
Yes po,
Are you
madam.
here? Ikaw
Yes
na
po.
po.
Sige po, madam.
Yes po.
Si Carms po ang mag-present po ng PPT ko. Wait lang po, madam. Wait lang po, madam. Okay na po.
Nice.
So, ang ginawa ko lang po dito, sinamarize ko lang po yung day 1 to 3 to 4 to 7. So, on day 1 to 3, in the light area setups, only 4 setups have a fuzzy light and smooth modes, with the majority of them being wet light setups. In the dark area naman, 6 setups have modes, which is composed of 3 wet dark setups and 3 dry dark setups. However, the mode covers a wide area in the bread on wet dark setups than the dry dark setup. On day 4 to 7 naman, some wet light setups are almost covered with modes, whereas the dry light setup have modes on the side of the bread only. In the dark area setup, all of the wet dark setup are covered with molds, and same with the light area setup. The molds in the dry dark are growing and spreading from the side of the bread slowly. So in my own conclusion po, mold grows more quickly on wet bread than it does on dry bread because mold thrives in moist environments. But it grows up. Molds can grow in both light and dark environments, but it grows faster in the dark since molds are not photosynthetic and doesn't use light to generate energy. Yun lang po. Thank you.
Eventually, kapag nag-progress na ang ating mga lesson, pasunod-sunod, dinadahan-dahan ko para sa inyo lahat. We will know bakit sila nag-grow ng mas mabilis sa light at bakit mas mabilis sa dark. Okay po? Sige. Thank you for that, Franz. Next is Bray. Brian? Wala din si Bray?
Andito ma'am, wait lang po.
Okay. Ayun nga pala. Sorry, Bray. Nasa unahan ka pa pala. Hindi ko ma-turn on ang aking camera.
Nailing na po, madam.
Yun na tabi, Bray.
So, good morning everyone.
Good afternoon, Bray.
Good afternoon. So, this is my presentation about my bread mold experiment. So, first of all, let's first have a short introduction about molds. So, molds are also spelled as MOULD. In biology, is a conspicuous mass of mycelium or masses of vegetative filaments or hyphae and fruiting structures produced by various fungi from the kingdom of fungi. So, here are my results. So in my results, I have it in the time period of day 1 to 3, all of the wet dark setups, specifically bread 1, 2, and 3 showed growth of mold. In day 2, breads 1 and 3 already showed signs of growth. And on the next day, bread 2 followed, showing fuzzy hair-like structures of mold, while the other setups did not show any signs of growth of mold. So ayan po, nakikita po dito sa pictures, yung bread number 1 is almost fully covered na ng mold, tapos yung sa number 2, nag-grow pa lang. Tapos, on days 4 to 7 of the experiment, bread number 5 that came from the light wet setup showed signs of growth of small amount of molds. The same bread, like number 2, it growed fuzzy hair-like structures out of bread number 5. And in dark wet dark wet breads, which is the breads 1, 2, and 3, have already revealed a lot of mold development, with bread number 1 being covered with dark mold, leaving only spots of white bread visible. In bread number 2, the dark mold seems to have attached the bread. Perhaps, parang nag-form siya ma'am ng parang breach ng hair-like structures dun sa cup nung Tupperware. Tapos,
O yun, spread na masyado ang iyong spread na masyado
Yep,
yung mycelium. Opo.
tapos yung number 2, parang nag-attach siya sa Tupperware tapos hindi na siya magalaw.
Okay.
Tapos in bread number 3, it had shown like, yun na nga po. Tapos, in conclusion, molds grow in a fast rate in the dark places, especially in wet environments, rather than in dry and bright environments. Molds thrive in damp and humid and wet conditions because they require to grow and spread. So, which is why it is recommended to keep our home dry as possible. Although bread number 5, that was from the wetland setup grew mold, maybe yung reason doon is that nung days 4 to 7, makulimlim yung panahon, talagang hindi man lang naarawan yung
Mold.
setup. Opo. Yun lang po, ma'am. Thank you.
Okay, so salamat for that. You have to remember that hindi lang light, hindi lang moisture ang nakaka-apekto ng growth and development ng ating mga fungal mycelium and for them to develop fruiting bodies. Nakaka-apekto din ang temperature. Isang malaking bagay ang temperature para mag-control ng growth and development ng ating mga fungi. Sa mga setups in laboratories, kadalasan nilalagay sa refrigerator ang mga setup. Ito ay nilalagay sa refrigerator para kahit papano ay mas slow down ang growth ng mold. So, sa malamig na lugar, nag-grow din naman siya, but hindi siya basta-basta nag-i-spread ang kanyang mga mycelium. But kapag mainit-init ng konti, not naman on the extreme hotness na temperature, ay hindi rin naman ulit siya mag-grow. So, sa mga susunod na araw, ito po ang ating pag-uusapan tungkol sa paano sila nag-grow at paano nag-develop. So, iba-ibang klase ng mga fungi, basidiomycetes and other species ay magkakaiba-iba ang growth development. Okay, last one is Erica. Erica, ikaw na po.
Good afternoon po, ma'am, and everyone. Ito po yung conduct compare. Yung observation ko po with the light, with yung bread na may light and dry. Yung fungi po, it started na mag-appear noong day 5 with thin, smooth white spot and mostly found on the sides of the bread. And then, nung day 7 naman po, it was found with smooth white spots with grayish bumps and yung top and side na po ng bread yung na ng fungi. With the light and wet bread naman po, nag-start po yung appearance ng fungi on day 4 with smooth white spots and mostly found on the side and some of the top on ng bread po. And when I experiment, there are white spots and mostly nasa side po ng bread and may mga grayish bumps of fungi po found sa side and konti po sa taas. And then, observation naman po sa without light and dry bread. Yung fungi po started na mag-appear with on day 5 po with light, smooth, white spots and mostly found po sa taas ng bread. And at the end of the experiment po, few grayish bumps po ng fungi ang nakita sa sides and sa top. And may mga white spots naman din po ng fungi. For the bread without light and wet, yung fungi po started to appear on day 4 with smooth white spots and mostly found on the sides of the bread and few on top. And yung naman po, end ng experiment on day 7 may mga grayish bumps po ng fungi and few on top, and white fungi are found on the side where yung fungi po nag-spread na po kahit po sa container sa taas po. So, eto po. So, ang conclusion po, based on the experiment that we have conducted, I have observed that once po yung fungi nag-start na to grow, it would rapidly increase day by day, and I have also discovered that molds might have the same, as nag-increase po yung mold, it might have the same color and texture, or it might be new one na iba na po yung color and texture. And during the experiment po, I have discovered that fungi does grow in a dark and wet environment, pero it doesn't rule out po the possibility na mag-grow pa rin po yung fungi sa dry and yung may light source po. And then yung moisture po sa environment ng bread, it also affects yung pag-increase po ng production ng mold. Ayan po.
Okay, thank you for that, Erika. Nakita nyo, magkakaiba-iba rin naman. Halos pare-parehas ang results nyo, but magkakaiba-iba kung ano ang mga growth patterns. And as we have mentioned last time, matinding effect yung amount ng water na nilagay mo. Kapag tamang-tama lang ang moisture na naandun, ay talagang nag-grow siya ng sobra-sobrang maayos. But kung sobra-sobra naman yung water na the bread gets to be flooded with water, hindi naman siya basta, hindi nakakapag-spread basta-basta ang ating mycelium. Okay, so thank you for that, everybody. Meron lang tayong dalawa na hindi nakapag-submit at nakapag-report ng kanilang results, si Phil at si Angela. Hindi ko alam kung anong mga rason. Hindi ko alam kung ayok. Yes po. Yes po. Francis.
Ako pa po, ma'am, present pa po.
Yes. Yes, ikaw nga ang last.
Yes po.
Francis.
Si Dina po mag-present ng screen.
Okay, Dinah, present na po ng screen ni Francis. Ang ganda naman yung mga picture na road. Ang tagal lang mag-load.
Amo na tayo na.
Okay.
Start.
Dinah mo na. Opo.
Hello, this is the summary of my experiment, bread mold activity. Actually, ma'am, day 12 talaga po ini lahat. Pero yung first na 3 days, wala po talaga kahit sa wet sample po nagtubo na molds. Kaya nag-start po grabe po ako sa day 1 po. So yung common
Okay.
color po na napansin ko yung green, white, and yung orange, tapos eventually nagiging brown po siya and yellow. Yung texture naman po, commonly yung fluffy sa sample 4, the smooth, as fuzzy po siya sample 11. Pero yung sa sample 11 po siya, ma'am, nagkagwa po siyang mold sa day 5, pero after day 6 hanggang day 9, nawala po siya kasi parang na sobrang po yung water dito. Next na po.
Okay.
Here is my final observations. All wet breads begin to moist on the first and second days while don't have changes as observed. The most successful samples to grow bread molds are the wet samples 4, 5, and 6 at dark area. Dark area setup has a colder temperature than light area. That's why molds grow faster. Light area disadvantage might be its high temperature because molds don't and cannot grow in a high temperature. All wet bread samples moisturized at the first 2 days, while the dry bread became flat. To prevent mold growth, check the expiry date of the bread. Some containers should be tightly closed or sealed. Keeping the bread in a dark place may not be helpful to prevent the growth of molds. Um, the water, a good substrate, the bread, and temperature and light um, might be helpful to grow molds. In water, mold thrives in moist, humid, and wet environment. Water is required for their growth and dispersion. In good substrate naman po, the amount of moisture in bread has a big impact on how quickly it molds. While temperature and light, the molds grow at warm temperature because the proteins that contribute to the growth of molds work more efficiently in higher temperature than lower temperature. My conclusion po,
Yes.
ay, sorry. This activity, bread mold experiment, gives data and information about how molds grow in bread samples and what benefits and drawbacks they have when placed in a specific setup. Too much water, exposure to light, and improperly sealed containers are all disadvantages, whereas it is favorable to inspect the bread sample for high bread quality, close the containers appropriately, and place it in a comfortable environment. Furthermore, as we delve deeper into the experiment, we notice that the size, texture, and color of the molds alter as the day progresses. That's all po.
Yes, thank you for that, Francis. Very good. Babago-bago ang color. And Francis made mention about optimum temperature. True. Sa mga susunod na lesson, meron po tayong lesson on that. Okay? Sige. Salamat everybody. Meron pa po bang hindi pa natawag? Anybody? Na hindi pa natawag? Wala na? Okay. So, we continue the lessons. Please post your reports and then your final paper on the in Google Classroom. Parehas ko na po siya na edit kung kailan magsasubmit. So, wait lang. Dali lang. Bread mold. Okay, bread mold experiment. Wait lang po ha. Tingnan ko lang kung na-edit ko yung edit. March 4. So, yung mga ngayon po ay pwede pang mag-submit ng inyong mga final paper. Nagawa nyo na rin naman po siguro. So, mamaya din po hanggang, sige, basta ngayong araw na to. Okay, sige. Edit na rin po. Please, Okay, so I proceed now to the next lesson. I gave you already the PowerPoint of this last meeting. And I hope you get to browse. Kung hindi man nag-browse, ay makinig po ng bonggang-bongga ngayong hapon na to dahil mabibigat na po ang mga susunod na mga lesson. So, nakikita po ang PowerPoint. Can you see the PowerPoint? I also asked you last time to watch the Fungi Kingdom Fantastic Fungi video. If you watch the Fantastic Fungi video, kung hindi nyo pa na papanood yung pinadala ni Cor na link, ay mamanood po kayo dahil sa mga susunod na araw ay kasabay na ang mga yon sa ating lesson. So, ngayong araw na to, pag-uusapan natin ang characteristic ng fungi, paano nag-evolve ang ating mga fungi, paano yung classification, at ano ang mga fungal life cycles. Ang fungal life cycles na pag-uusapan natin ngayon ay pahapyaw lamang or just an introduction because later on we will be talking more about this per clique ng ating mga fungi. Kung paano sila nag-disperse ng kanilang mga spores, paano sila nag-i-spread out ang kanilang mga mycelium at paano sila gumagawa ng kanilang mga fruiting bodies para doon ang kanilang mga spores ma-produce. So magandang hapon, ang pangalan ay Kingdom Funga. This is a new coined name of the kingdom, although hindi pa naman siya ganun ka-popular, but just to describe the fungi as a kingdom, ang tawag na po ay funga. Nakikita po. Habayaan ko ulit ang pagbalik-balik. Nakikita po. Ay.
Yung editing lang po, madam.
Okay, yan po. Nakikita po. Ha?
Yung editing
Hello?
lang po, madam.
Ito, nakikita na?
Hindi po. Yung ano lang po, yung editing lang po, madam.
Ay, Lord. Ito po, nakikita?
Hindi
Yes?
po, madam.
My God. Wait lang. I-share ko na lang ang buong ano. What's wrong with ano?
Madam, share mo po yung window po ng habang nagpe-present ka po. I-present mo na po.
Sinishare ko na po. Sinare ko na ang ano, stop presenting muna. Share ko na lang ang buong ano ko. Buong, tawag dito? Okay po. Share na lang ang buong screen. Buwis itong Google Classroom na ito sa totoo lang. Share entire screen. Share. Share. Okay? Kita na?
Hindi pa po
Oh,
nag-share.
Lord. Bakit ayaw yata tayo? Bakit pag Biyernes palaging ganito? Kahapon naman sa ano ay hindi naman ganito. Pag Friday palaging ganito. Okay, share. Screen. Share
Wala
na?
pa po, madam. Yan na po, may ano, yan na, okay na po.
Okay na?
Yes po, madam.
Yan. Ha? Can you see?
Yes po.
Okay. So, magandang hapon again. As I mentioned earlier, ang pangalan po ay Kingdom Funga. And pag-uusapan natin ngayong hapon na to ang characteristics ng fungi, ang evolution, ang fungal classification, at ang fungal life cycles. Ang lahat ng ito is just introductory for today. Sa mga susunod na araw ay hihimay-himayin ang bawat characteristics ng fungi at paano siya nakaklasify at ang kanyang mga life cycle. So, nasabi na natin ito last meeting. Ang kanyang body form ay unicellular. And they are filamentous or tube-like. At ang tawag sa tube-like strands na ito ay hypha for a singular strand or hyphae for plural strand. At kapag pinagsama-sama ang napakaraming hyphae na nakita nyo na fuzzy at you describe it, ang tawag na po doon ay mycelium. At kung nakikita nyo, ang inyong mga mycelium ay talagang nag-spread ng sobra-sobra, ang iba na puno na hanggang doon na nag-colonize siya sa buong lalagyan ng inyong bread. So, ang tawag po doon ay aggregate of hypha at ang tawag doon ay mycelium. At meron po siyang palaging ginagawa na nag-harden up ang mga mass ng mycelium natin. At ito ay nagsiserve as an overwintering stage at ang tawag po dito ay sclerosium. Dun sa ibang mga pictures na pinadala nyo sa akin ay merong mga parang mga bola-bola na sobra-sobrang maliliit na wala pong mga stalk. At ang tawag po dito sa matitigas na bola nito are sclerosium. These are hardened mass of mycelium na nag-overwinter. Pag sinabi po nating nag-overwinter, para po siyang nagka-hibernate. Hindi siya basta in extreme conditions kapag naitapon mo siya or biglang naging super dry ang environment ay ito po ay bato ay bilog-bilog lang at hindi na parang hindi siya mag-grow. But when the conditions becomes favorable, merong water at merong tamang temperature for growth and development, ang mga sclerosium po na ito ay magsisimula mag-grow ng mga hypha and eventually would aggregate again into a mycelium. So, nakikita nyo itong picture na ito. If you can see this, ito po ang nailagay po dito sa gitna nito ay isa pong sclerosium. Paminsan ang tawag natin dito ay sclerosal bodies. Kung kayo po ay observant sa inyong mga gardens, makikita nyo na parang may mga seeds doon sa soil ng inyong mga plants. At ang akala, naghihintay ka na tumubo ang mga ito. So ito po ay mga sclerotial bodies, at ang sclerotial bodies is an aggregate at nag-harden ng mga hypha or nag-aggregate mycelium na nagtigas at nagbuo ng isang bola. And of course, they are multicellular such as mycelial cords, rhizomorphs, and fruit bodies. At yung nakita nyo na may mga stalk, sila po ay multicellular na at sila po ay mga fruiting bodies na nakita natin, at ang pinakamalaking example nito are the mushrooms na nakikita natin at kinakain po ang iba. So ito po ang isang basic characteristic ng ating mga fungi. So ito po ay isang example ng fungi na meron pong fruiting body, at kadalasan nakikita natin ito sa mga basidiomycetes. Yung karamihan ng mga basidiomycetes ay nagpo-form po ng mga ganitong structure na ang tawag na natin ay mushroom in general, at ito po ay mga fruiting bodies. At kung nakikita nyo po sa picture na ito po sa baba na picture, nakita nyo yung mga mycelium nag-i-spread, and eventually nakikita po natin yung mga fruiting bodies na merong mga cups, ang iba merong mga gills, ang iba naman ay merong mga pores. Eventually pag-uusapan natin. But just to show you kung anong itsura ng fruiting body at ano ang laman ng fruiting body, ito po ang laman ng fruiting body in a cross-sectional dimension. So makikita mo yung mga mycelium extends from the substrate kung saan siya naandun. Pwedeng sa kahoy, pwedeng sa mga leaves nakadikit-dikit, at ang fruiting bodies ay ang mycelium extends towards the fruiting bodies. And eventually, dun po sa mga fruiting bodies, dun po natin makikita dito, nakikita niyo yung mga gills, dun po natin makukuha ang mga spores that gets to be spread out para mag-continue ng existence ng species na yon. Okay, so sabi natin last time also, hindi siya nagpo-photosynthesize. At this particular group of organisms are heterotrophic in nature. Heterotrophic dahil sila po ay kumukuha ng kanilang kailangan na pagkain sa nature, at hindi nila kaya mag-photosynthesize kagaya po ng mga plants. Sa fungi classification, nung una, sabi ko sa inyo, sila po ay nakaklasify kasabay ng mga plants because of their structure. But eventually, dahil nga sila ay heterotrophic, they were classified again na malapit sa animals. And but eventually, sa pag-progress ng biology, sila po ay hiniwalay at ginawa po silang isang kingdom, and now they belong to kingdom funga. And anong klaseng heterotrophic mechanism for food getting ang meron sa fungi? Una, saprophytes, or they are saprobes. So they feed on dead tissues or organic waste. At ito na mga saprophytes or saprophobic fungi plays a big role in decomposing materials in nature. Sila din po ay symbionts, which is actually a relationship na mutually beneficial, like a relationship between a fungus and another organism. At ang example po natin ito nung isa are the lichens and the mycorrhizal fungi, at sila po ay nagbibigay ng benefit, mutual benefit, at nakakakuha rin po sila ng benefit sa kanila pong mga host. And of course, they are also considered as parasites. They feed on living tissue of a host. Marami pong mga fungal diseases ang nakikita natin sa iba't ibang mga organisms, sa plants and sa animals, and they feed on the living tissue of their particular hosts. And ang mga parasites po na fungi na nakakapag-cause ng disease are also called as pathogens, kagaya po ng mga bacteria at ang iba pang mga organisms na nakakapag-cause ng disease on other organisms or sa kanila pong mga hosts. Okay, so paano po sila nagkumakain? Heterotrophic din po ang mga animals, but ang mga fungi po ay kakaiba. Ang kanilang being heterotrophic po is by absorption. So fungi get carbon from organic sources. Paano nila kinukuha? Yung kanilang mga hyphal tips ay nagre-release po ng enzyme At ang mga enzyme po na ito ay nagbe-breakdown ng substrate at yung mga products na naandun po sa kanilang mga substrate ay nagdi-diffuse back papunta sa kanyang hyphae. Wait lang po. Wait lang! Wait lang po ha. Sandali lang po ha. Yung mga yaya ko po tulog kasi. Wait lang po. Give me 3 minutes. Rachel? Rachel? Ayan na, Sally Aisa. Yes, Sally Aisa. Pasensya everybody. Yung mga yaya bells ko, pag nagpatak na ang alauna, ayan, natulog na. Okay. Saan ako sabi ko? Ano sabi ko? So the products because of the enzyme nadadigest nila yung mga substrate kung saan sila kumukuha ng pagkain, and this particular byproducts of the process of digestion using enzymes are the products diffused back into the hyphae. So, tingnan niyo po itong picture. Oh, yun. So, maglabas siya ng enzyme. Halimbawa, nandun siya sa mga leaves, maglabas siya ng enzyme. Okay. Yun. So, nagkakaroon po ng enzymatic breakdown. Pag nagkaroon ng enzymatic breakdown na digest, at yung na-digest na yun ay ina-absorb po nila. At sila ay nandun. And just the same mechanism with plants and animals, yung kanila pong mga nucleus ang nagdidirect ng lahat ng activity, kasabay po yung paano magre-release ng enzyme, anong enzyme ang i-release, at paano mag-a-absorb back ng maraming bagay. So, ito na yung byproduct out of the enzymatic activity. So, the product diffuses back into the hypha and is used by the hypha for growth and development hanggang sila ay mag-aggregate into mycelium at maraming. So, paano natin i-characterize ang hypha, ang hyphae or ang hypha kung isa lang? Sila po ay tubular. Sila po ay merong hard wall of chitin, made up of chitin. At meron pong mga cross walls and may form compartments. Cross walls, halimbawa po ganito. At meron ding compartment ito. And ang ibang species are ang karamihan sa mga species are multinucleate, but there are also some species who are uninucleate, but ang karamihan basically ng mga hypha are multinucleate. Ibig sabihin, ang dami-daming nucleus. Kasi nga po, ang growth ay naandun sa tips at elongate lang at nag-replicate na ang at nagkaroon na ng maraming nucleus. And the hyphae grow from their tips at ang tawag sa eventually, sabi natin kanina na nakita nyo, are the mycelium, yung nag-spread out na fusy material na sabi ninyo, and they become extensive. As nakita nyo sa inyong bread, sila po ay nag-spread at na-colonize ang buong bread. At kung makikita natin, ito yung feeding web ng mga hyphae. So lahat ng materials na naandun sa substrate na pwede niyang i-digest ay i-absorb niya. And mycelia are the ecologically active bodies of fungi. So, hindi na lang hypha ang sinasabi natin dahil nga sa mabilis po sila mag-grow at mabilis po ang kanilang development. At ang nakikita nyo na maraming mapuputi at mga fuzzy na sinasabi nyo na mga bagay-bagay dun sa inyong setup are the mycelia. Balikan ko yun. Okay. Okay? So, eventually, yung kanyang tip ay parang plastic na nag-i-stretch. But ang kanyang wall naman ay very rigid. So, dito lang po ang growth and development niya sa unahan at wala pong growth and development dun po sa mga sides dahil nga sabi natin kanina, meron po silang chitin. So, this particular picture shows you modifications ng mga hypha. So, paminsan-minsan, ang hypha ay lalo na dun sa mga fungal species na nagpe-predate or na nagiging parasitic in nature sa kanilang mode of feeding, sila po ay gumagawa ng hostorium. Ito po na mga hostorium na ito ay para pong mga ugat, ay para pong mga roots. And basically, this particular hostorium impregnates the cell of a particular plant body or plant species, at ito po ang nagkukuha ng lahat ng materials doon sa loob ng plant cell. Okay? So siya po ay naandun na sa may plasma membrane as nakikita nindo dito, at napapasok niya ang plant cell wall, at ayan na po siya. So it gets to be anchored into the plant cell and utilizes the whatever is in the plant plasma membrane para sa kanyang nutrition. So pwede pong dalawang bagay, pwede pong septate at pwede pong non-septate. Paminsan po yung walang septa, ibig sabihin wala pong mga boundaries. Boundaries, ang tawag po ay sinusitic. Dahil po yung kanyang mga nuclei ay nakakapag-free flow dun sa hypha or sa mycelium. But sa iba naman po, ang iba namang hypha are actually septate. So, isa-isa lang ang mga hindi nakakapag-free flow or move out yung mga nuclei or yung mga nucleus na naandun sa kanya. So, this is an example of a hyphae adapted for trapping and killing a particular prey. So, ito po ay isang nematode, and this particular species of fungi actually predates on the nematode. So, yan. Ito po yung hypha, at siya po ay nag-i-encircle by producing hostorium na parang root system that anchors the hypha or the fungal body into the nematode, and thereby killing the prey or the nematode afterwards dahil naubos niya na kung ano ang naandun sa nematode. Okay. So, fungi are also considered as saprobes and decomposers. At ito ang inyong bread, yung paraan ng pagko-colonize actually nung mold nyo dun sa inyong bread ay paraan para mag-decompose ng bread. So, ito rin pong species na ito is a wood decay fungi. So, ang dinidecompose niya rin po ay ang wood kung saan siya nakakabit. And of course, sa ating mga fruits, and pwede rin po tayong nung cultured na mga oyster mushroom, meron pong tinatawag na meron pong mga bag na meron pong mga laman. Paminsan po ang laman ay sawdust. Paminsan naman po ay, ano ba ang tawag niyo doon sa rice hull? At doon po siya nag-grow. Ito. At sa mga bahay-bahay natin, nakikita natin paminsan, lalo na kapag very damp at ulan ng ulan, ay meron pong nag-grow na mga maitim-itim sa ating mga ceiling, sa ating mga floors, sa mga gilid-gilid, and sa mga walls. At ito po are saprobic and decomposing fungi. And of course, sabi natin, they are symbionts, and this is a mutualistic relationship. Yung wood decaying fungi, of course, sinasabi pa rin palagi mutualistic. Bakit siya mutualistic? Dahil yung nakapaligid doon sa wood decaying fungi, nabibigyan niya rin ng nabibigyan niya ng benefit. Kapag siya ay nag-digest nung wood decaying fungi, ay nung wood, yung isang wood decaying fungi ay nabibigay niya at nababalik niya doon sa mga nakakapagbigay siya ng pagkain sa kung ano-ano mga organisms na naandun pang iba. And of course, they act as mycorrhiza, pwedeng sa loob ng roots ng mga plants, lalo na ng mga trees, or pwede namang sa lapa. So, either it can be endomycorrhiza or ectomycorrhiza. And of course, ang pinakamaganda ay ang ating lichens. At isang example also of a symbiotic relationship is semot or the black yeast, which actually exists on the stomach of a particular species of ant. Ano ang ginagawa niya? Anong tulong ang nabibigay niya dun sa ant? Ang nabibigay niya na tulong is nahi-help niya mag-digest yung ant ng kung ano-anong pinakain. So, we give emphasis on the mycorrhiza. Bakit? Dahil ito ay fungus in the roots. And ang mutualistic pattern between the plant and the fungi is that the fungus ay nagbibigay ng nutrient at nagtutulong ng water uptake ng plant dahil naandun sila sa roots. Ang plants naman, dahil nga sa nag-distribute siya sa kanyang phloem ng mga materials that have been produced in photosynthesis like carbohydrates, sila po rin ay nagpo-provide sa fungus dun sa root system ng mga carbohydrates.